Hi, I wanted to briefly introduce the chromosome 18 conditions because it can get a little complicated. Let me explain. So, here's a picture of chromosomes taken under a microscope. And imagine if you had this picture and a copy machine and scissors and glue. How many different combinations could you create from this picture and multiple copies? You could cut here, there, everywhere. Anything you could do to this picture with scissors, glue, and a copy machine can really happen, and it does really happen. It's just that so many of them are not compatible with life, and babies with those combinations are miscarried, usually very early in pregnancy. But some of the conditions that are viable to birth and associated with a long life are the chromosome 18 conditions. So I'd like to describe to you what they are. And now that you know that there's huge variety and lots of combinations and lots of variability, now I can tell you that we try to put those into 5 different groups. So you know that each group is going to have some variability within the group, but we put them into 5 different groups. The first group is by far the most common. It's trisomy 18. Most of these babies don't make it to their first birthday, and so they tend to be very medically fragile. And there are also other organizations interested in trisomy 18 and other research groups interested in trisomy 18. So we've not focused on trisomy 18 except to try to enroll the long-term survivors over age 4. We're very interested in that group because those individuals tend to be more like the rest of our cohort with other chromosome 18 abnormalities. They're not so medically fragile anymore, and we're very interested in how to, what challenges they have and how to improve their lives going forward. The next group is 18q minus. So individuals with deletions of 18q are rare. They occur in about 1 in 55,000 births. And a deletion of someone with 18q minus has a deletion from 1 copy of chromosome 18. So 1 copy is normal, but the other copy has a deletion of any size from any region within this long arm of chromosome 18. And you can see when you look at the chromosomes that they all have some sort of like a waistband. It's called a centromere. So they all have a short arm and a long arm because a centromere is never exactly in the middle. And just the convention is that chromosomes are shown when we show pictures of them and drawings of them with the short arm at the top. So 18q minus is a deletion of any size from any portion of the long arm. So you can imagine that is highly variable. And it's in fact so variable that we have enrolled well over 300 individuals, and no 2 people have exactly the same deletion. But what we have found is there's a region in the middle of the long arm of chromosome 18 that nobody has deleted. It's right here shown by the red arrow. And what that does is that makes 2 groups of people with 18q deletions. Those with distal, so farther from the centromere, they have deletions in this region here. And people with proximal, so closer to the centromere, people with deletions in this region here. The proximal deletions are more rare. It's only 17% of our cohort, where the distal are 83%. And the proximal region has 83 genes, and the distal region has 98 genes. Then there's tetrasomy 18p. This is very rare. It only occurs in 1 in 625,000 births. And these individuals have 2 normal copies of chromosome 18, but they have an extra chromosome. And that extra chromosome is made up of 2 p arms. So they have 1, 2, 3, 4 p arms, and so we call it tetrasomy 18p. And most of these people have exactly the same duplication. There's only a couple of people who have a little q arm material, but most of them have the entire P arm duplicated. So that group is pretty homogeneous. Then there's 18P minus, and these individuals have a deletion of any size from any region on the short arm, the P arm of chromosome 18. And that occurs in 1 in 56,000 births. So again, pretty rare. Then there's ring 18. So ring 18 is very interesting because individuals have one intact copy of chromosome 18 and their other chromosome 18 forms a ring. Now in order to form a ring, both ends of the chromosome have to be missing. There are caps on the ends called telomeres, and when those are missing, then the ends are sticky and they can stick together and form a ring. So by definition, there's missing material from both ends of the chromosome, but there's not necessarily genes missing from both ends of the chromosome. So you can imagine this group of people is very heterogeneous because they can have different amounts of material missing from both ends of the chromosome. And this occurs in about 1 in 300,000 births. So just to talk about or explain some of the terminology that you see when people talk about chromosome rearrangements and deletions. There are people who have terminal deletions. So here's an image showing a terminal deletion. So one chromosome is normal and this chromosome has this, the very end of it is missing, and that's called a terminal deletion. And people with deletions of the long arm, the end of the long arm, so distal 18q minus, can also have interstitial deletions. So here's an image showing a hypothetical interstitial deletion. As you can see, this region right here shown in the bracket is missing in the chromosome with the deletion. So this band is here and that band is here, and what's in between them is missing on this chromosome. So that's a distal interstitial deletion. But you can also have an interstitial deletion of the proximal region. In fact, those are all interstitial deletions because you can see this band is here and this band is up here, and so this region in the middle is missing from this chromosome. So this is a proximal interstitial deletion. And to make it even more complicated, sometimes people have, in addition to their deletion, a duplication. So in this case, in fact, this one is only a duplication. This dark band right here is duplicated over here. So this person has an interstitial duplication of 18q. And in this case, this person has a terminal deletion. So this region is missing from this chromosome here. And this region here, this dark band, is duplicated. And in fact, many of the people with terminal deletions, so distal terminal deletions, have a little bit, not anywhere near as big as shown here, but a little bit of a duplication right at what you'd think of as the healing point of the abnormal chromosome. So you may see that in the literature or described in your reports as well. On top of all that, there's also mosaicism. So this happens when there are two or more than one cell type in the body. So in this cartoon, each of these little hexagons is supposed to represent a cell. And the blue ones have normal chromosomes. The yellow ones in this case have tetrasomy 18p. So this person, if this were someone's cells, let's say a skin sample, there are 25 normal chromosomes and 5 with tetrasomy 18p. So 5 out of the 30, or 17% mosaicism is what's shown here. But people can have two different cell types where both cell type is abnormal, or they can have mosaicism where one cell type is normal and one is abnormal. So mosaicism is quite rare. It's only a couple percent in most of the conditions, except for ring 18, where I think about half have mosaicism of some sort. So that's more common in that group, but that's another term that you may hear. So, given all that, you can see that anything you could do to this picture with scissors, glue, and a copy machine can really happen, and it does really happen. But in order to try to make sense of it and to help families think about it and find people most like their child, we have grouped these conditions into these 5 groups. And so I hope that helps to explain some of the terminology that we use when we talk about the chromosome 18 conditions. And I would be remiss if I didn't point out that these gorgeous pictures of these amazing kids were taken by Rick Guidotti of Positive Exposure. So, thank you so much.